name of our project. Our research has really focused based on Jackie's memory on the particular type of leukemia that she was diagnosed with, which is this MLL leukemia. We've been doing a genomic analysis of patient samples who have MLL leukemia. Leukemia, for those of you who don't realize this, is a cancer of the blood. And it's a situation where the normal blood cells have gone bad. They refuse to differentiate into normal white blood cells. They get stuck at a very early stage of development, and then they overgrow. In fact, they overgrow to such a high level that patients like Jackie can present with 10 or 20 times the number of white cells as a normal individual. The site that the leukemia begins in is the bone marrow. The leukemia cells, as well as normal cells, grow within the bone marrow, and then they go out into the blood. And you can see here that normal bone marrow, and on the bottom, is full of different types of cells at different types of growth and differentiation. In a leukemia patient, the dark blue cells, which you see in the bottom left-hand corner, are the leukemia cells. They're very large, they grow out of control, and they rapidly spread throughout the body. Because it's in your blood, it's not limited to just one location. It's throughout your body and the blood at the time of your diagnosis. Each of our cells, normal cells and cancer cells, contains DNA. DNA is a genetic code that you inherit from your parents. The DNA determines your individuality as well as your similarities that you have with both of your parents. DNA is arranged in structures called chromosomes, and each chromosome consists of thousands of genes. The genes are the parts of the chromosome in the DNA that actually tell cells how to function. And for example, the genes on the X and Y chromosome determine what gender you are. Now, the particular type of gene that defines the leukemia that we're studying is the mixed lineage leukemia gene. And this gene is a gene that's found on junctions or break-off points of chromosomes 4 and 11. When the 4 and 11 translocations break off abnormally, they reform and they link portions of chromosome 4 with chromosome 11 in an abnormal way. The transfer of genes from chromosome 4 to 11 to form a new gene, the MLL gene, leads to abnormal gene function. The abnormal gene functions that result from these abnormal portions of 4 and 11 are what actually lead to changes in the normal blood cells that leads to leukemia. So the processes by which these cells become leukemia is directly linked to this MLL gene. That causes these cells to grow out of control, allows them to renew, and for them to be resistant to many types of chemotherapy drugs. And so when you look at acute leukemias that have an MLL gene, as opposed to leukemias who do not have an MLL gene, the patients whose disease is characterized by an MLL gene have a much poorer survival. They do not respond as well to chemotherapy, and when they do respond to chemotherapy and go into remission, they have a much higher chance of going back out of remission and having the disease recur. So in this particular graph, if you look on the y-axis, that's the percentage of patients who live without leukemia. And you can see that if you have a leukemia that is not characterized by an MLL gene, your overall survival is about 70% of those patients will be able to live long-term with standard chemotherapy. If you have a leukemia with an MLL gene rearrangement, your chance of living following standard therapy drops down to about 20 or 30%. Okay, so MLL leukemias are really characterized by poor outcomes and irresponsiveness to standard chemotherapy. And that's typical of what we see in our patients that we treat here at Roswell Park and in other cancer centers. Now, the goal of our project was to identify the genes and pathways that are involved, not just the MLL gene, but what the MLL gene does to the cells to make them so resistant to chemotherapy and make them such poor responders when we give them chemotherapy. Maybe we shouldn't be giving them chemotherapy. If chemotherapy is only curing 20 or 30% of them, maybe we should develop a better therapy for these types of leukemias with this gene that works better than chemotherapy does, okay? And in order to do that, it's good for us to understand what are the biological processes resulting from this MLL gene problem that leads to this type of cancer. And that's really what we're looking at. What are the genes? What are the biological pathways? And can we use our information to develop better therapies? So the way we decided to study this, Dr. Nowak and I, was to say, well, we don't really want to study leukemia in chimpanzees or frogs or fruit flies. We really want to study this disease in actual human patient samples because that's the best way for us to understand the disease. So what we did is we went to our tissue bank here at Roswell Park. Every time we see a leukemia patient and we make a diagnosis of leukemia, we try to make whatever good we can of a bad situation, and we take some of the leukemia cells from our patients and we ask them whether it's possible for us to save some of them. So what we've done here is we have freezers full of cells that we've saved from our leukemia patients that have been treated and diagnosed here at Roswell Park, that were material that they didn't use, but that they said that we could save for future research purposes. What we've done is we've identified 28 patients who had MLL-associated acute leukemias here at Roswell Park. There were 14 women, 14 men. We looked at their clinical situation. We looked at samples from patients at the time of their diagnosis, at the time they went into remission, at the time that their disease recurred. And we decided to study these samples to see what information we could get from the patients who were treated here that allowed us to store their material. The way we decided to do this, and we discussed this a little bit last year, is with their advances in technology, we have the ability to study every single gene in every single cell. So for example, there are 33,000 genes that we have identified, and we're able to take cells. In this particular case, we take the tumor cells. We extract the RNA and the DNA from the tumor cells, and we take them and we're able to coat them on sort of plates or arrays that are studied with certain reagents specific for each of the 33,000 genes and all of the genetic abnormalities and things like that. We are then able to put them in these sheets or arrays into machines, and the machines will scan all of the different genes that are on that particular plate and are able to tell us, is that gene upregulated, is that gene downregulated, is the abnormality present, is the abnormality not present. And what happens is, then you get an image of this sheet of paper where there's all these different highlights on the sheet, depending on whether the gene is upregulated or downregulated, and then the computer is able to analyze those plates and give us a whole list of information. So what it looks like is like this. You have these arrays with all of these dots, and each of these dots represents one gene expression. And you can see that this looks like, very difficult to interpret, and we use our computer analysis to make sense of all of these dots. In a similar way, we're able to look at cytogenetic or chromosomal aberrations in all of these patient samples, and we're able to look at genetic modifiers and microRNAs. And what happens is you just get all of this information that looks like this or like this. And really, when you're looking at this, this is what you're saying. What does what does all of this data mean? And this particular lecturer says, well, I'll pause for a minute so you can let this information sink in. But really, it doesn't really, in this type of format, make any sense to any of us. And so in terms of what we're doing with this data, we need to be asking questions that are going to allow us to analyze that data and make some sense out of those thousands and thousands of genes. So one of the questions that we've been looking at and we've asked initially is, when we look at acute leukemias and we look at a patient like Jackie, there are different situations in which leukemias can develop in patients. One situation that can occur is you can have a healthy individual who one day develops a cancer. There's no risk factors, there's nothing genetically, nobody else in the family has a history of cancer. It's something that the cancer develops spontaneously. And for those particular patients, we say that they have a primary leukemia. We also have patients that have something wrong with their bone marrow or their blood system for many, many years. And over time, those particular disorders can become leukemic because the bone marrow gets more and more damaged. Those patients, we decide, are called secondary leukemias, that they have a first problem and then they develop a second leukemia. The third situation that people can develop leukemia is if you develop breast cancer or lymphoma or colon cancer and you receive chemotherapy or radiation for your prostate cancer. What the chemotherapy does is it damages your blood cells. and what patients who have received prior chemotherapy or radiation can develop a leukemia from the damage to their blood cells from a prior chemotherapy or radiation injury. And this can occur in up to 10 or 20% of patients that are treated for other cancers. And so what we asked ourselves is, when we look at these MLL leukemias are responsible for about a third of therapy-related cancers. So when you look at the risk factors for leukemia, you can talk about things like, when you look at risk factors for cancer, smoking, diet, exposure to toxins, radiation. These are all things that can cause leukemia but in our therapy-related ones, the difference is that these patients have received some kind of chemotherapy or radiation. And what are the differences between MLL leukemias that occur in individuals that don't have any of these risk factors, like a spontaneous leukemia, and those that develop therapy-related? Being able to look at what is the common mechanisms between those two types of cancers and what are the differences can give us a lot of information about how they're different. Why do people spontaneously develop leukemia? Why do people develop leukemia in other situations? So one of the questions that Norma and I asked was, can we identify differences in the types of samples that we're looking at? Can we determine what makes a young individual's cancer different from an older individual's cancer that got prior therapy? How does that information tell us how these leukemias develop? And how does that give us information about how to target these specific types of leukemias? And so this is just an example of some of the data that we've been able to generate. The red and the blue represent different expression of different genes. In this situation, we have 305 genes that are summarized by this red and blue kind of mess here. You can see that when you look at these 305 genes, if you group all the samples from patients who had a therapy-related leukemia and those who had leukemia with no other prior risk factors, you can see clear patterns. So I think you can see this, so that in the therapy-related samples, all of the genes on the left side are more blue, and then on the right side they're all red, whereas in the primary leukemias it's the opposite. So there's clearly genes that are up and down regulated in specific patterns that are related to the type of leukemia that they develop. And when we tried to graph this in a different way by using sort of different types of analyses, so here what we do is we take each of the leukemias and we plot it on this graph depending on the expression of these genes as a dot. So instead of looking at that big blur, you just look at the dot. You can see here that all of the blue dots are on this side and all of the red dots are on that side. And that again tells us from these patient samples that these two diseases are very, very different biologically. They're dependent on different processes. And I don't think nobody has ever shown that in MLL leukemias, that there's a difference between the type of leukemia that you get when you're a primary leukemia and then when you're therapy-related. Now, what do these genes represent? And when we looked at what these genes represent, we can see there's a lot of biological processes that are involved in these genes. Things that differ between these patients are differences in their immune system, in their response to stress, even things, inflammatory responses. All of these processes that we've identified that are different between these two types of leukemia are very basic biological processes. So these are real things that we've identified in these patients. And we look at some of the processes that some of the genes are involved in, we can see that some of the differences in these leukemias are due to things like even differences in DNA repair or chromosomal packaging, genetic things that have to do with the packaging of the genes and the DNA. These are differences that we see. It may be that some of the leukemias, the therapy-related leukemias, result from DNA damage, whereas in the spontaneous leukemias, those are processes that aren't as important. It may be that the processes that go on in a young patient develops an MLL leukemia are very different than an older patient. So we are starting to look at some of these specific signaling pathways that are different. And graphically, when we look at them, you can see, and I don't have a pointer here, that there are differences in some of the processes that have to do with the DNA packaging, some of the processes that have to do with receptors, some of the processes that have to do with interactions between the leukemia cells and other immune cells. So these are some of the basic biological processes that I think that we've been able to now focus in on in our research and identify as being things that are wrong with these types of leukemia cells. We've also been able to identify specific genes are different between these two. And the reason that it's important to find a specific gene is once you have a specific gene, you can look at inhibitors of that gene. You can look at therapies that target that gene. You can knock down that gene, and you can look at that in the laboratory and see whether that would represent a potential target for therapy-related versus primary leukemias. And some of the genes that we've identified already, we know, are genes that are responsible for chemotherapy resistance. Remember I said that the MLO leukemias are very resistant to chemotherapy. We're starting to dissect that process by identifying the genes that are responsible for that. So I'm very excited by this data. I think that we've been able to really focus in on some targets, and we're starting to sort of unravel things a little bit in this last year. What are we doing now? Well, there's not an end to this story. What we're doing is we've identified another 13 samples that we'd like to look at. So in addition to the 28, we have about almost another 40% of patient samples we've been able to accumulate, and those samples have been processed and are in the process of being analyzed. Obviously, the more samples that we get, the better we'll be able to confirm our data and we'll be able to refine our data. We also had a teleconference with somebody from the Mayo Clinic who we've requested to collaborate with, who may have dozens of more MLL leukemia samples that we would have the ability to study. And we have told them that they give us the samples that we, with the Jackie for ALL Foundation, we would have the resources to be able to study those samples to really make this a comprehensive story and to allow us to extend our collaboration to MLL patients at other cancer sites. So we're very enthusiastic about that. The genes that we're looking at, we're hopeful that we're going to confirm those genes, and then we would move into ways to target them or inhibit them to see whether they really would represent new therapies for patients. And given our information right now that our data is very novel, nobody has shown any of this data for the MLL leukemias in patient samples. We're optimistic that with these additional 13 samples that we may have enough information to submit our work for publication in the early part of 2012. And that publication will have Jackie's name on it, and will acknowledge the support of the foundation that have made this work possible. We will then take that publication and use it to apply for NIH grant dollars, which in this day and age are very rare, but I think that the data is really coming to fruition now, and we will be starting to see Jackie's name on some academic publications and grant proposals in the next 6 months. And our goal eventually is to take some of these targets, look at them, target them in cancer cells, target them in mice that have the leukemias, and from that point, when we're able to move our data from identification of the genes into certain gene targets and then into sort of clinical trials in mice and in cancer cells, I think that's the next step forward. And I think that over the next year or two, we hope to have more information of this sort, targeting information, identification information, so carry the project forward. Once again, none of this work would be possible without Jackie and her presence in our lives. Although she's not here, I'm sure she is looking down and is very proud of what has come. And we thank her and her family and all of you for all the efforts that you've made to keep her spirit alive and to move this research forward. The dancers give back. You guys are phenomenal. In this day and age where they say people don't care, they're not caring, they're only self-centered, they only care about themselves, it's not true. And you guys are an inspiration moving forward to future generations of dancers and for the dancing